So if you guys haven't done so already, make sure you answer the question in the link that Armin sent about if adapters were detected or not. I'll give another second or two. If you guys lost the link, accidentally closed out, Armin, maybe you could put the information in the chat one more time just in case. And I'm going to go ahead and go over the answers, which I think you guys all got right, it looks like. Let's see. Next one here. Okay. Yes. Everyone said yes. Indeed. That is correct. Okay. Were adapters detected? Yes, they were. How do we know? Did I pass already? Adapter content right here. So this graph here, we see that literally every single sample has adapters. So indeed, adapters were detected. And then the last and final question, Armin, if you want to put this one up, should we trim and or filter the data before aligning to the reference genome, and why or why not?
So, it should be up.
Oh, awesome. Go ahead and fill in your response to that question. And I'll give you guys probably just a minute before we go over this. I do want to make sure we get through our trimming section before the end of the day. Go ahead and put in your answers. Should we trim or filter these data based on what you guys noticed about quality, about adapters, what we went over in the lecture? Do you think that we should do some trimming or filtering of these data?
We've got 3 answers so far.
Guys, do we trim or filter these data? Why or why not?
All right, we have 10 answers.
Okay. Should be almost everyone. So in the interest of time, I'm going to go ahead and show those results.
There's 11.
Awesome. Okay. Yes, yes, yes. Trimming the data will help with many things that could potentially go wrong. For one, it would remove low quality reads. It would also help with trimming out the adapters. It would also remove reads that were too short. Yes. What are guesses? The guesses, trim adapters. RNA contaminant, those few ends at the beginning, we need to filter out smaller reads too. Yes, we should. Trim and filter out adapter content, as well as put length limit, decrease change of mapping. Sequences are just right. Let's go for it without trimming. I'm curious. So there are some bioinformaticists that are just like, we should never trim. So it's true that some don't like to trim at all. Personally, I do think that we should trim. I think you should always do a quality filter and always remove adapters because when we go back to map, anything that didn't come from our samples, like if we added something artificially like adapters, I think that we should remove those, right? Because those aren't part of biology. There might be a chance that, I mean, hopefully depending on how the adapters were designed, hopefully they won't map to a certain location, but we don't know that for sure. So to minimize off-target effects or minimize false information, anything that we add artificially, like adapters, we should definitely remove before we align back. Since these data are really high quality, I mean, a quality trim isn't gonna probably do much to change anything because they are, where's that graph? Higher. Because they are really high quality. And if you guys remember from the lecture, we trim anything that is less than 20 for quality. What would be the reasoning not to trim? We're going to get into that when we go over alignment. But the reason is depending on how you set your alignment criteria, you can account for a certain number of mismatches. We also, depending on what type of aligner you use, if you use a splice-aware aligner, if there's part of the read that does not map to a certain location in the reference genome, it can assume that it is a splice isoform. And so because things can account for that and look at like parts of reads to align, some people argue that that's a reason why. I disagree with those people, Susie. So I think that we should always remove anything that we added artificially. But you will get some folks saying that aligners are really smart nowadays and we can get away with it. But I disagree. So yeah, so we're probably not going to see too much with a quality trim, right? Because all these are really high quality. And as I mentioned, we trim anything less than a Q score of 20, and we see that none of these have a Q score of 20. However, because as many of you noted, we do see those adapters, that we do want to get rid of those adapters because, again, we added those on. How do I keep missing the graph? We added those on artificially. And so, there they are. We added those on artificially, and so I really think that we should get rid of anything that we added artificially because it didn't come from our samples. To the note, I know someone really wanted to be aggressive with their trimming and even trim at that 5' end to get rid of that little blip that we see here. Some people do trim at the 5' end. I wouldn't just because, again, depending on how you set your alignment criteria, how we set it, as we'll go over next week, is we allow for a 4% error rate. And so if we have 4 mismatches out of 100 base pair long sequence, we'll still accept that mapped. And so because I only see those ends at like the first or the second position, and it's in such a few number of reads that it's not going to affect the mapping really to have those ends there. So I wouldn't trim the 5' end, and at that point, depending on how deep you trim at your 5', if you just lop off 1 base pair, not too much, but like some people will do some intense trimming and trim off like the first 5 or 10 base pairs, trim off from the 3' end a bunch. So we do trim, but we do fairly conservative trimming, so we don't go crazy. So we will do our quality trim, and we'll get rid of adapters. So that's our next step. So we are moving on now to trimming, just to remind you guys where we are in the workflow. I scroll up the top here. So I know it doesn't seem like we've gone that far. Things are going to pick up. These fast QC reports, we could probably do a whole boot camp just on those. I always learn something new when we go over them again. So we have assessed the quality of our raw reads. So now we're going to, now that we saw that we have adapters in there, we want to at least trim for adapter. We're going to do the default quality trimming too. And so we do that with a tool called Trim Galore, which uses under the hood another tool called Cut Adapt to do the trimming. And so I know we went over this in the lecture. So just as a reminder here, these are the parameters that we use. We use Trim Galore, so we're going to go over these parameters in the notebook for this next step. If you guys scrolled up like I did, we're at part 2A now. So to trim and filter our raw sequence data with Trim Galore. So again, to ensure maximum sequence alignment to the reference genome, our next step is going to be to clean up the raw sequence data using Trim Galore. Again, just like I did with multi-QC, this will link out to the Trim Galore documentation in case you guys want to read more about it. And as I mentioned, it utilizes another software, Cut Adapt, kind of under the hood there. So if you want to learn more about Cut Adapt, which is the tool that's doing the actual trimming, you can learn more about it by clicking that link. So during this step, we filter the raw reads to remove any sequencing adapters detected, which of course were artificially added to enable sequencing of sample RNA. And then we also get rid of those low quality base pairs. So I do have this question here. I think this might be a question. I think I kind of harped on this, but does anyone, Armin, is this the next question? It might be, I'm not sure.
I don't think I added that one.
Okay, good. Yeah, so I kind of went over this already, but any idea why we would want to do this before aligning to the reference genome? If anyone wants to unmute their mic or put it in the chat.
To limit the chance that smaller sequences will be placed into multiple different locations?
So remember, right now we have, what are our sequence lengths right now? This is,
Okay.
So what's our current sequence length of our raw data?
128 base pairs.
Where'd you get 128?
Oh, I'm sorry. I read the wrong thing. Sorry
Yeah,
about that.
if you take a look at the sequence length distribution, this is a little weird, so I'm kind of glad you brought this up. Because you remember the length for the reads for raw data should all be the same. Are these all the same? From this graph? No, yeah, they're not. So what lengths do we see here? There's two lengths. Give you a hint. What are the two lengths that we're seeing? Yeah, Ty, that's right. So we are seeing that in all of our samples, we either have a length of 100 base pairs or we have reads with a length of 150 base pairs. How could this be if these are raw data and we set our sequencer to sequence at a particular number of cycles, which will give us the specific read length that we have? Well, I'll tell you because I didn't go over this. One thing is we could say, oh, maybe these aren't actually raw data and some trimming already happened, and that's why we have some that are 100 and some that are 150. Well, I could see why you would think that, but it would be really odd if after we trimmed, we only got two exact lengths for all of our reads like we have here. So what actually happened here is these samples were sequenced before we had a sequencer at GeneLab. So we actually outsourced these samples to have sequenced at UC Davis sequencing samples. And we had them sequenced and we told them the read depth that we wanted per sample, and we wanted to sequence it originally at paired end 100 base pairs. So they sequenced all of these samples at paired end 100 base pair and gave us back our data. This was before I joined GeneLab. And then when the bioinformaticists that were here at the time took a look at those data, they noticed that they didn't have the total number of reads for every sample that we had requested. And so we went back to UC Davis and said, hey, you didn't give us the total number of reads per sample that we wanted. And so they said, okay, we'll sequence deeper. And so they took our samples back and sequenced them again. But it turns out that they added it with another group's samples, and the other group samples were sequencing at 150 base pairs long, and they just kind of added our samples onto that sequencing run and gave us back our data, and the additional reads that they had added were 150 base pairs instead of 100 base pairs. So, no, that's no problem, Mark. I'm glad questions are good. And so what happened was we had now some sequences for our samples. Most of them were 100 base pairs from the initial sequencing run that they went through. And then the subsequent sequencing run that they went through, they were sequenced at 150 base pairs. Now, normally, this would be an issue. We wouldn't want to see this, but because if you noticed, there's roughly the same number of reads that were sequenced at 150 for every single sample, and roughly the same number of reads that were sequenced at 100 base pairs for every single sample. So it's kind of like, how when you make an error in your wet lab, but you make it to every single sample, but when you're doing a comparative analysis, it kind of cancels out. Similar thing. So because we have a mix of roughly the same number of reads at 100 base pairs and roughly the same number of reads at 150 base pairs for every single sample, we decided just to keep all the reads and go with it. So Again, this is uncommon. You should see with your raw data just one read length for every single read. So good thing to point out the weird data. So we want to go ahead and trim. And so Mark, to your point, once we start trimming, then we're going to start to see a variation, as we'll notice in the read length based on the quality of each read and how many base pairs were adapters in each read. And so we're going to trim our forward and our reverse read for all 12 of our samples using trim galore. And that command is set up as follows. So this is the same command that I showed you guys in the lecture. So we are going to run the trim galore command. This is how it's called. So this tool is called trim galore. This is how the tool is called. Again, you'll know that because if you Google the documentation or click on this link to go to trim galore, you'll know how to run the command or do trim galore -- help. A couple of options here. All the parameter definitions, again, you could pull up the help menu to know what these are, but I did point out the parameter definitions of the parameters that we use. So this first one, this gzip option here, tells it that when you give me back the FASTQ files that have all the reads that have been trimmed, I want you to gzip those files, so to compress them. -- cores, this just specifies the number of cores to do the trimming on. This has to do with the capability of the system that you are doing the trimming on. For red 33, that's going to tell the CutAdapt tool to use the OSI2 characters, OSI233 characters for the quality scores. So when it's interpreting those quality scores in the fourth line of each read, what letters it's used to indicate the quality scores there. -- Illumina is telling the tool to use the Illumina adapter sequence. It's letting them know, hey, the adapter sequences that you're going to find here is from Illumina. And so it has embedded within the tool the Illumina Universal Adapter. Note here that it starts with an A, AG, AGA are the first three base pairs. That's going to be important when we interpret the FASTQC data. We have -- paired, so this indicates paired end reads. So this is really important when it's doing that final filtering. So that is where Mark is going to do the filtering based on how long the reads are. So this is where it's going to get rid of any read that's less than 20 base pairs after trimming. And the fact that we have this paired option means that if either the forward or the reverse have been trimmed so that they're less than 20, both the forward and the reverse read will go away for that specific read. Because you have to make sure that the number of forward and the number of reverse reads are the same. And then we have this -- output dir, which is the option to indicate where to output the FASTQ files and the trimming reports. And then this last thing here is, again, a positional argument. You notice that there's no -- before it. So trim galore just wants you to put the files that you're going to be trimming, so your raw FASTQ files, as the very last part of the command. So at the very last part of the command here, we have our forward and our reverse FASTQ file. And again, you see a similar setup for when we're running our bash script for FASTQC. For the raw data, we're holding it in this special variable with the curly brackets because we are going to process all of these with an array script, because again, it's going to be the same exact command run. The only thing that's going to change are the samples that we are giving it for an input. And so we're able to do that with the slurm array script, as we saw in great detail for raw FASTQC. So those are all the parameter definitions. The input data that it needs those raw reads. So these are the input data that we are providing it, is the raw reads for each sample. It does have to be in this format, so forward read first, a space, and then a reverse read again at the end of the command. The output data that we expect to get from this tool is our trimmed reads. So these are going to be the trimmed reads, so it's the remaining sequence data after quality trimming, after adapter trimming, and after it filters for read length. We're also going to get a trimming report, and this is going to give us information about how the data were trimmed and filtered. So this is going to tell us what happened to our data during this trimming that we've performed. And so we can go ahead and run this command. Again, I have a little note up here that the trim galore command can take over an hour to run per sample. And so this, like we did with the FastQC, we're going to submit to the Slurm job scheduler and allow it to run in the background, although this is simulated, so we're not actually running things. But this is what it would look like if we were to submit to a job scheduler. So you guys can go ahead and run that. And you should see an indication that your job was submitted, and you should get a job ID. And so while that's pretending to run, we're going to take a look at these output files. So again, 2 output files we expect here, well, 2 sets of output files. One is our trimmed fastq.gz, and we should have a trimmed forward read and a trimmed reverse read for each sample. And we should also get 1 trimming report for each sample. Sorry, 2 trimming reports, 1 for each file, forward and reverse for each sample. And so we're going to take a look at those trimming reports. So first we're going to take a look at the trimming report for the forward read, and then we'll take a look at the trimming report for the reverse read for one of our samples. So again, we're looking at this flight rep 1 sample here. So you can go ahead and run that. And this is what the trimming report looks like. So after you're done doing your trimming and your quality filtering, you'll get something like this in the trimming report. So what you'll see here is kind of a summary of what happened. So it'll tell you what file it worked on. So again, this is our rep 1 of our set of flight samples, and this is the forward read. Trimming mode, so this is basically regurgitating the settings that you gave it, so you can make sure that you did indeed put all the settings that you wanted to. So paired N, these are paired N data. It tells us the version of both trim galore and cut adapt, because remember, this is actually running another tool called cut adapt under the hood. Couldn't detect a Python version, that's okay. Number of cores used for trimming. So this again is number of cores have to do with the capability of your compute system. So we set 2 cores to be used. The Phred score quality cutoff. So this is our Q score cutoff. And if you remember from the lecture, we use the default cutoff, which is 20. So any base pair that has a quality score of 20 or less will be removed. The quality encoding type, so this is what those quality scores are encoded by, so the SE233 encode, and just a quick reminder if this person has seen it, so that those are these scores that I went over in the lecture, so that are of course in that 4th line of our read, of each one of our read. So it basically is going to take this code and convert it to the Q score that is respective of its code. And then if that Q score is less than 20, and it takes into account the base pairs around it as well, and it'll remove. It also tells you what adapter sequence it's looking for. So again, this is the Illumina Universal Adapter. The maximum trimming error rate is 0.1. So this is a default error rate. So basically when it's looking for an adapter sequence, it allows a 0.1 error rate for identifying the adapters. The minimum required adapter overlap industry is 1 base pair. So this again is a default. That means that if it detects so much as 1 base pair that matches up with this adapter sequence, it will get rid of it. So this is important because that's saying if it sees an A at the end, it's going to get rid of it because it's going to match up to the adapter here. Minimum required sequence length for both reads for a sequence pair to get removed, so 20 base pairs. So this is that read length cutoff. So again, we use the default of 20 base pairs. So after it trims off based on quality, then it'll do adapter trimming. And then when it's done trimming, it will remove anything that's 20 base pairs or less. But I have a question about when it actually does the removal, which we'll talk about. And then it's just reminding you the output files are going to be gzip compressed, which again, we set in the parameters. So this is just tell you it's moving. So it does tell you what Python version. I don't know why I couldn't detect it up there. But then it tells you the version of Cutadapt, version of Python, more about the parameters and shortcuts, where it processed the read on, and then how long it took. So this program actually will calculate the total time. And then here's the summary of what it found. So the total reads processed, 68,373,507. How does one undo gzip? Good question. You could do a tool called gunzip. So that'll unzip it into standard out. So unzip is if a file is just zipped. So if you have a file that ends in .zip, you would uncompress that on the command line with unzip, I think. Someone correct me if I'm wrong. If you have a gzipped file, you do gunzip to tell it that it's compressed using the gzip format, and we want to uncompress it. So gunzip will uncompress it. If you want to view it without actually compressing it, which we could do if you guys want to, you use a sister of the cat command called zcat, and that will open up a gzipped file without actually printing out without actually uncompressing the file. So the file will remain compressed. You'll just be able to take a picture of what's inside of it without fully uncompressing it. Reads with adapters. Now it tells me, so this is the total number of reads for this sample, right? So this actually should match in the FastQC report. If we were to go back up, right, this is the flight rep 1 sample that we took a look at up above. So we had 68,373,507 reads. That should be the total number of raw reads that were there. But just to prove that to you guys. If you remember, raw 1, total number, 68,373,507, right? So it should function on every single read in that file, which indeed it does. And then it tells you how many of those 68 million reads had adapters. So 22 million, over 22 million of them had adapters. And then it tells you what percentage that makes up. So about 32% of the reads for this sample had adapters on them. And this is the reads written. So this is passing the filter. So after quality trimming, after adapter trimming, this is how many reads were outputted. So here we have 68,370,507. And then again, it gives you the percentage. So 100% of reads passed were large enough after all the quality filtering to continue on. We're going to have a little more when we look at the reverse. And then it gives you this number in base pairs. So then it tells you the total number of base pairs, right? So here we're looking at full reads. Here we're looking at just base pairs. So, each read has 100 to 150 because of the dichotomy that I told you about the sequencing issues with this data set. So it should be, 100 plus however many were 150 reads times the number of million reads that we have to give you the number of base pairs. And then it tells you of the total number of base pairs, how many base pairs were quality trimmed, and then again in percentage, so 0.1% of all base pairs were trimmed, and then how many total base pairs were there when filtered. So it gave back 98.6% of the base pairs from the original. More information from the trimming report. So again, it tells you the sequence. This is the Illumina universal adapter. And how long is length 13? It says to trim from the 3' end, and then it gives you a summary of how many times that was trimmed. So that I should match this, yeah. So how many times it was trimmed, that's the number of reads that it was trimmed on. Number of allowed errors. Allowed errors meaning mismatches between the universal adapter and what it's reading in the read. And so, right, sequencing, as we know, isn't necessarily perfect. There can be base pair mismatches. And so this is just saying we use the default here. We didn't actually set this, so we use the default. So by default, it won't allow any errors in the first 9 base pairs. So the first 9 base pairs of the sequence have to match exactly in order for it to be detected as in order for it to be detected as an adapter. Longer, 10 to 13, it will allow up to one mismatch from the adapter and still recognize it as an adapter and remove it. It tells you the percent of the different base pairs that came just before the adapters that were removed. And then this is just a whole bunch of information about the removed sequences based on the length of the sequence that was removed. And so there were 13 million, a little over 13 million reads that had a length of 1 that was removed. There are over, I'm just in here, 4 million reads that had 2 base pairs removed, 13 million reads that had 3 base pairs removed, etc. on down. This is if it was randomly finding a base pair that would match the adapter, what it would expect to remove just based on random. The max error is the max error allowed. So really this length of 1, the fact that it has a max error here, it should be 0, right? So this is based on this, the number of errors allowed. So really things that were cut off at 1, in the first 0 to 9 base pairs, it shouldn't have that. So if it does, it indicates that there was actually like a deletion that was detected. Probably too much information for you guys. You don't really have to pay attention to that.
I think that's just shifted over, Amanda, the zeros for your
Okay, that makes more sense.
max error. Yeah.
That does happen though.
Yeah.
I was just reading it up. If there is a deletion, that will be weird. But yeah, thanks, Mike. So this is just tabbed over. Sorry. So this did have a 0, right? So really in your first 8, well, 9 technically, right? We don't allow any errors. So those really should all be 0 unless there's a deletion that's detected. So it's smart enough to know if there's a deletion or not. And then we can see in now a length of 10, we have at least, we can allow up to 1 error, right? So 10 to 13 base pair length of the adapter, right? The adapter is 13 base pairs long. So it's saying no errors in the first 9 base pairs, and then base pairs 10 to 13, it'll allow up to 1 mismatch. So this is saying with the length of 10, there might have been detected 1 mismatch. And then here is the number of reads that contain a length of 10 that was removed that had no errors, so matched exactly with the adapter and then those 10 base pairs. And this is the number of reads that had a length of 10 base pairs that were removed in which one of those 10 base pairs didn't match exactly with the adapter. So again, really more information than you guys probably need to know. But this was a question when we were doing a dry run of this that people really wanted to know what these columns were. So that's a summary of what those columns are. The things that I really pay attention to here that are most important in these trimming reports is really kind of the total number of reads processed, total reads with adapter, and the number of reads that were written out. And there's a region that the forward read prints out all of them. Even though if you notice, this is, right, so this is the length that was removed. And so you'll notice that some of them had the full length removed, 40 had the full length of the read removed. So you should have presumably gotten rid of some reads. Why do you think that they didn't remove any reads after trimming the forward read? Why wouldn't they remove any yet? Any guesses? This is paired end data. Remember what I told you about what's needed with the total number of reads in forward and reverse for paired end data. And right now it just went through and trimmed the forward read. So why do you think, even though we see here that there were some reads that had a lot of adapters or quality that was removed, why would it not have gotten rid of the read yet? No one wants to guess? Fear of unanswered. It's okay, this is all learning. This is the first time you guys are learning about this stuff.
Could you repeat the question one more time, please?
Yeah, so if you notice here, right, this is telling you how many reads of a particular length were removed, right? So we know that these reads in total are either 100 or 150 base pairs, right? From the FastQC analysis of the raw data. So for each of these samples of the reads are either 100 base pair or they're up to 150 base pairs. And we know that the cutoff that we gave trimming to completely remove a read is if we trimmed it so much that there was only fewer than 20 base pairs remaining, that you would just get rid of that read, right? And so we see here that there were at least some level of reads that had over, so if we're letting it be 20 base pairs or higher, keep. So that means that if it had to remove at least 130, right, since our longest read is 150 base pair. And we see that there are reads, if you add up all of these numbers here, that's just this column of numbers here, that's how many reads had to remove 130 or more base pairs, which means it only had, if it started with 150, it only had 20 or less left. So there were reads that ended up being shorter than 20, right, after this quality trimming and adapter filtering. However, if you look at the summary up here, it said that the reads that were written out that passed filters was 100%. So in the forward read, it gave us back all of our reads according to this summary. Why would it not have gotten rid of them?
Because isn't it just between 100 base pairs and 150 base pairs? So wait, no, okay, I caught myself in the logic. I'm not sure. I got to think about it some more. Sorry.
So right, you have something that's up to 150 base pairs, right, the read, and you're saying, okay, this quality's crap, I'm cutting that off, I'm cutting that off, I'm cutting that off as a quality. Oh, now I see some adapters, I'm cutting that off, I'm cutting that off, I'm cutting that off. And it cuts off so much that it gets to having less than 20 base pairs left, right? We know that happens if we look at here, because we see, again, this is the total length that was cut and the number of reads that had that length that was cut. So, right, so one read in this sample had 139 base pairs that were removed from the read, that were cut from the read, right? So that means that if it started at 150 and it cut off 139, then that's less than 20 that was left. So if it was less than 20 that was left and that was our cutoff, you would think that it would remove that read, right? Because it ultimately was less than 20 after doing the filtering. Exactly, Joel. So what we're seeing here in this summary is that the total number of reads that it started with is the same as the total number of reads that it gave back to us.
So it's just the trimming. It's saying it trimmed all that out, and it gives us all those lines back, regardless of whether or not it met the criteria of having whatever more than 20 amino acids. I mean, base pairs.
Right. Protein guy.
Yeah.
Why would that happen for the forward read? Remember, this is paired end data, and we know something special about paired end data. What does the forward and the reverse read, the number of reads in the forward and the reverse, have to be in paired end data?
The same length?
It has to be the same length. So now does it make sense why it has not removed reads yet? We're just looking at the forward right now.
Oh, because then we need to go and pass that against the backwards, and then we'll do the same filtering, and then we'll mash them together at the end, and then see if any of it actually marries up.
Exactly. So right now,
There it is.
that these are all forward reads that we're looking at, but we told it that this is paired end data. So when you tell TrimGalore or ultimately CutAdapt that this is paired end data, it'll go through and it'll trim all your reads, all your forward reads first, but it's not going to remove any reads, even if they don't meet the criteria yet. It's going to wait until it does the filtering for all the reverse reads. And then after it's done the filtering for all the forward reads and all of the respective reverse reads, then it'll see, okay, for each read, I should say for each insert, right, that we sequence in the forward and the reverse, how many base pairs are left in that forward read compared to that respective reverse read. And if either one, if the forward read or the reverse read is less than 20, it gets rid of both the forward and the reverse read. Does that make sense to people? Is everyone getting that?
When we're doing that, like if we're removing opposite ends of each read, so like if we're left with 20 on the far end and 20 at the front end, or 21 on either end, what does it do then in that case? Because they're not perfectly going to marry up, are they? Like when we align reverse to forward.
It can handle that. Yeah, that's fine.
Okay,
So,
okay,
yep. So yeah, and that's true, and that does happen, right? So,
because that's it. So when you're talking about the contig thing, I never understood why contigs was such an important thing in alignments, and now it makes sense why it would make sense. Yeah, okay, yeah, all good. I think I'm getting it now. Whoa.
awesome. So yeah, so we can have different lengths of the forward and the reverse, right? That's fine. But we have to have a forward and a reverse read. And so that's why, even though some of the forward reads didn't meet the 20 base pair cutoff criteria for the read length, they're all still there after we look at the forward read, because Trim Galore is going to wait and do the trimming on both the forward and the reverse read before it does a length cutoff.
Yeah, I have a problem with this whole thing.
Which problem?
Trim Galore Cut Adapt is writing out the first log file, yes, but it is processing them both at the same time as far as we're concerned. Like if we look at the read one file, there are fewer reads than the total. So really, it's giving out false information for the first read file. It never writes out a file that has the full set of reads with empty lines. So I have a problem with the way their documentation works, and people have complained about this before. But I also don't like that you made this a question, like it's logical to do it the way that they're doing it, because I don't think it is at all.
That's fair. Yeah, that is fair. Because it's basically telling you that your end product for your FASTQ file has for your forward file that has all the reads printed out, which is not going to be
Right, and if you count it, they're not all there. That's not the number of reads written.
right. It's basically the number of reads that it has stored in memory until it finishes.
Exactly.
Yeah.
Yeah.
Fair. Good point.
Anyway, minor point. No reason for me to say that, except I couldn't keep my mouth shut any longer about it.
No, totally understand. Yeah, so now that it's gone through and as Mike pointed out, it has this information kept in memory, that it still has all of the reads there in memory again, because it's waiting until it does the trimming for read 2. And so now we're going to look at the read 2 trimming report. So we can go ahead and run this next command. So now this is the trimming report for the same exact sample, but the reverse read. And so again, we see summary statistics. So the input file name, trimming mode being paired N, version of Trim Galore, version of Cut Adapt, number of cores, quality Fred score cutoff, the type of coding for the quality scores that's used, the ASCII-33, the adapter sequence that we're looking to trim off, the max error rate that we allow for the adapters, the fact that we need only one base pair to match the adapter for it to cut off, and then the length that it has to be at the end of the day. Again, both the forward and the reverse have to be 20 base pairs or more in order to keep it. And then again, telling it to gzip those files that are produced, the FASTQ files that are given back to us. So that summarizes kind of the same thing here. And then now we see here, and this does the same thing, Mike, to annoy you. So now it's holding all of this in memory, and it's telling you how many reads were processed, how many reads had adapters. So I want you to notice this might be a question that I'll give you the answer to, but note the number of reads with adapters in the reverse and in the forward. It's always interesting to me. And the percentage, it's a little bit more than the forward. And then it says that they all pass filter again, because again, it's running read 1, it's running read 2, and printing out this summary, but it gives it all back. And then after it does this, that's when it's going through and taking a look at the final lengths of each respective read for forward and reverse before it does the final cutoff. So again, this tells us that it's giving back 100% of the reads. And we can see that broken down by base pairs now. So here it's broken down by read, here it's broken down by base pairs. So how many base pairs were quality trimmed, and then total written back out to us for base pairs. and then the number of errors that we allow when we're trying to identify this adapter sequence in our read. And then we see here the percentage of each base that preceded the adapters that were removed. And then we have the summary again of the length, how many reads with that length were removed, how many it would be based on random error, how many errors it identified, and then a count of the number of reads with each number of errors that it identified. Again, errors meaning differences compared with what the adapter sequence is versus what it found in the read. So it goes through all that. And now we have a little bit different information, well, different additional information. So again, it tells us how many reads were processed in total at the end of this file. And then it tells us the total number of sequences analyzed for the sequence pair length validation. So this should be the total number of forward and the total number of reverse reads, and that should match, right? So they're both 68,373,507 reads in both the forward and the reverse. So that means that it analyzed the forward and the reverse that many reads. And now we have the number of sequence pairs removed because at least one read, at least either the forward or the reverse read, was shorter than the length cut off, which is 20 base pairs. So now we can see the number of reads that were cut off ultimately. And again, that read was cut off both the forward and the respective reverse read. And so I want you to go back through and take a look at the summary, and we're going to answer some questions. So we are running short on time, so I'm just going to give a minute to respond to each one of these questions. So again, your best guess, if you really don't know, please put that you don't know in the answer, so that way I know to go over it a little bit more in depth. So I'm going to actually use a timer and do 60 minutes. So Armin, if you want to release this question, how many forward reads were processed?
Yep, it should be up.
Excellent. So I'll give you guys a minute, up to a minute, to answer how many forward reads are processed. Armin, if you see that everyone answers before the minute's up, let me know. Because I won't wait if everyone already answered.
We've got 4 answers so far.
How many forward reads were processed? So basically, how many reads did they start with, and did they evaluate for trimming?
No, 8 answers.
Awesome. Got about 20 seconds.
There's 3 more of you.
Okay. That minute has gone by, so we're going to go over these answers together. Okay, you guys all got this right. At least everyone who answered. For those of you who didn't answer, how we find this information, how many forward reads were processed, we would scroll up to the report for the forward read, right? So it's up here, and we're looking at the forward read trimming report. And how we know how many reads were processed is it tells us right there at the top of the summary, total reads processed. Next question, Armin, this one should be quick and easy now that you guys know where to find this information. So the next question is going to be how many reverse reads were processed? So now you want to take a look at that reverse read trimming report.
It looks like I skipped that question and went to the next one.
Okay, that's fine. You guys want to put it in the chat? How many reverse reads were processed?
Yeah.
So we don't have that question set up. Yeah, Ty, that's right. Right. Yeah, Nisha's right. Like, right. Yeah, you guys got it. So yeah, the same exact number, right? And that makes sense because again, with paired end data, we always need to make sure that there's the same number of forward and same number of reverse reads. Is anyone still unsure or a little shaky on why there's the same number of forward and reverse reads? Okay. Yeah, so same thing, except this time we're going to look at the reverse trimming report. And we see here, if you go to the summary section of the report, total reads processed, 68,373,507. So the same exact as the forward reads, as it should be. Next question, which I think Armin probably already has up. Are the total number of forward and reverse reads processed the same or different? Should these numbers be the same and why? So I know I just asked this, so give another 60 seconds to go ahead and answer that one. Go ahead and answer it in the Mentimeter site.
Yep, we've got 5 so far.
I know we're getting towards the end of our first week. It's been a lot. Give you guys 15 more seconds.
And that's 9.
Great. 10 more seconds here. Okay. That was my timer. All right. Wrote the number, that's true. Yeah, these should be the same. It's the same number, the same. They should be the same. Is there the same amount of sequences? Just read forward and reverse. Yes, same. Yes, they should be the same. Same. R1 and R2 are paired. Yes. Same because it's paired. Same. Yes. Don't know why. Okay, we're going to go over why. They're the same and they should be the same because they're paired end reads. Each forward and reverse should have a reverse. Yes, paired end sequences. Same. Yeah, okay, so I'm just gonna quickly jump to here. I actually made this slide during the break for a different reason, but I guess I can use it here. So when we are doing the sequencing, right? So we have this one insert that's depicted here is the forward read, right, in one of the clusters. And so we're gonna sequence in the forward direction. You're going this way. And then we're going to sequence that same exact insert in the reverse direction. And so that's going to happen for each one of these clusters. So we're going to get one insert sequenced in the forward direction, and then that same insert sequenced in the reverse direction for each one of these clusters. And so the total number of clusters is going to represent the total number of inserts or sequences that we're sequencing, and we sequence it in both directions. So ultimately, what we do when we have our thing, if we have this in green here, is our insert, is the thing that we're sequencing, and we're going to sequence that same insert in both the forward direction and in the reverse direction. And so that's why, because we're sequencing each insert or each cluster, however you want to think about it, we're sequencing it in both directions. And so we should have the same exact number of forward reads as we have reverse reads, because the forward and the reverse is acting on just one insert. Does that make more sense of why we would have the same number of forward and reverse reads? Is anyone still confused on why we should have the same number? All right. So next, were any adapters detected in the forward or reverse reads? If yes, how many forward reads contained adapters and how many reverse reads contained adapters? And are these numbers the same or different?
So I split this question up into two as well because there was a word limit on the questions. I think the first one ends at forward reads, and then the next one will ask about reverse reads.
Okay, so I'll give you guys 2 minutes. After you submitted your first answer, go to the next question and submit your answer for that one, and I'll give you 2 minutes. Unless, of course, you guys all answer that more quickly. It's going to encourage you to look at the summary section of both the forward and reverse trimming reports, and it should tell you very clearly if adapters were detected, and if so, how many. Oh, can't submit the answer for the second question until the results are shown for the first. Okay.
Yes, I'm gonna switch it as soon as I get 10 answers.
Okay. Thanks, Armin.
Yep. Waiting on one more. There is 10.
Awesome. So now you guys should have access to that second question. We got about 50 seconds left, or if we get everyone to answer before then. Got about 30 seconds left for that second.
Yeah, we have 6 answers so far.
Okay. That's time, so we'll go ahead and view those answers. So the first question is, were any adapters detected in the forward or reverse reads? If yes, how many forward reads contained adapters? So this is forward. Most of you guys said 22,398,255. We got a couple that were different, so let's see how we find that answer. So again, we're going to scroll all the way up here to the forward read. So this is the forward read trimming report. And right in the summary here, it tells us reads with adapters and tells us the exact number of reads with adapters here are 22,398,255, which ultimately made up 32.8% of the total reads. Anyone not clear on how we got that answer? Okay. Moving on to the next one. So here, so I think the person that did the 24 million just read the wrong report. So all these are right. And so again, the report here for reverse, and again, we go to that same location, reads with adapters, and we see that there's 24,222,277 reads, or 35.4%, and they are different. And for me, I don't know if one of the other instructors can answer that, but that's always been confusing to me. This does happen, and I am not fully sure why. Let me go to the cluster conditions. Okay, right, because if we go here, and I'm going to go even further to our libraries, final libraries here. So, oh, I know why this is different. No, I don't. So I'm curious. Yeah, I do. Okay. Sorry, guys. Just figuring that out for myself now that I'm looking at this. And so what happens is we have this, this is our final library, right? And so we're going to sequence this by priming in this orange end here and priming in this gray end here, depending on if we're doing forward or reverse. And so when we do the sequences, remember if our insert is shorter than the number of cycles that we're sequencing, we're going to start to sequence into our adapters. And so we're going to sequence, if we're sequencing in this direction, let's say this is forward, so we're sequencing forward here, and then we're going to sequence into our P7 adapter, right? And keep in mind that depending on if it's single index or dual index, you're going to have an index on P5 and you're going to have an index on P7. So you will sequence into your adapter, but before you reach your adapter, it's going to be, well, it will be this here, which is the priming region, which counts as part of the adapter. But then also we're going to have a tag here, right? That index on, and we have a P5 and a P7 index if we do a dual index, which is what we do at GeneLab. And so it'll sequence into this adapter. The universal adapter is accounting for the sequence of the primer and the sequence of the region that's complementary to the flow cell. Because there's different sequences for your index, you're going to have a chunk of sequence that's 6 base pairs for us, a chunk of sequence that's your index that you could sequence into that isn't going to match up to your adapter sequence. And if it doesn't match up by more than 1 base pair, then it won't get identified as an adapter and it won't get removed. And that sequence for your index on your P7 is going to be different than the sequence. I know it's not here because this example is showing a single index, but another index here. And so that's why when it does the trimming, right? So again, sequence, in order for adapters to be there, your insert has to be shorter than the number of cycles you sequence. So you start to sequence into your adapter on either end. And that sequence on either end is going to be the same for the priming region and for the adapter, but it's going to be different for the index. And so because of whether or not you have your index on either side, you might not lose in how far you sequence into it. It might be slightly different forward and reverse. Does that make sense? Does that not make sense to anyone? Okay, so that's clear. So if that's good, we can move on to the next question. After adapters were trimmed from the forward reads, were any reads removed? So I don't think in the interest of time, and because we kind of really dove into this, I don't think we need to do the questionnaire. So maybe we could skip over this question, Armin. So we kind of really nailed down that no reads were removed after trimming the forward reads or after trimming the reverse reads initially, because it had to wait to trim both the forward and the reverse reads before it decides to remove reads. Because again, there has to be the same number of forward and same number of reverse reads. So it needs to wait and do the quality trimming on all the forward, do the trimming on all the reverse. And then once the trimming is done for the forward and done for the reverse, then it looks at the forward reads and its respective reverse read. And if either one of those is less than 20 base pairs, it'll remove that read. So we kind of really harped on that. So unless someone wants further explanation, maybe we could just move on to this one, Armin. So how many base pairs were removed due to poor quality in the forward reads, and what about the reverse reads?
Yep, that's the one that's up now.
Excellent. So I'll give you guys all a minute to answer that one.
I'm not seeing that question. I'm seeing the previous question.
Is that question
I
on?
think at the top you can click go to next slide or go to slide.
Oh, okay. Got it. Thank you.
Awesome. Thanks, Mike. Yeah. So keep in mind we have how many base pairs here were removed due to poor quality in the forward reads and how many base pairs were removed due to poor quality in the reverse reads. Five more seconds. Okay, I'm going to show answers. All right. So how many base pairs were removed due to poor quality in the forward reads? In the reverse, we're just over 5 million and then just over 18 million. And it looks like everyone who did answered that except for one person. So I think that you were responding to a different question, whoever put this number here. So yeah, how many base pairs were removed due to poor quality in the forward reads and the reverse reads? So you'd find that again in that summary section at the top of the trimming report. So in the forward read trimming report, I'll just double check, we see total base pairs processed, and now we see total base pairs that were quality trimmed. So those of you guys who put 5,291,484 base pairs for the forward, that was correct. And then if we scroll down and look at that same section in the reverse read, so down here, total base pairs that were quality trimmed, we have that 18,564,191. Does that make sense based on when we took a look at the quality of the reads? Yeah, Ty, it does. So remember, when we looked at the quality of the reads, we noticed that the quality of the reverse reads was worse than the quality of the forward reads, particularly at the end. And so that's reflected here. Excuse me, because we could see now that indeed more base pairs were quality trimmed in the reverse than the forward. Okay. Next question. After filtering base pairs for quality, what percentage of base pairs were removed? Oh, we just did that one. It's the same question asked twice. After filtering base pairs for quality, what percentage of base pairs were removed? Oh, this is asking a similar question. So in the interest of time, we'll skip that one too, just so we can do this next question before we end. So Armin, if you want to put up this next question. Did the raw forward or raw reverse reads have better quality? That one I think we covered pretty good too. Sorry, I know I'm skipping around just we have a minute left. So I'm just going to go over the answers to these questions. But after trimming and filtering, you guys put in the chat, did any read pairs fail to meet the 20 base pair length cutoff? If yes, how many? So I think someone wrote this answer already when I was asking about the base pair one. So pretty much how many reads were removed because either the forward or the reverse didn't meet the 20 base pair cutoff? You guys put your answers in the chat.
Somebody answered on Menti and said yes.
How many? You're right. I'm going to tell you because we're at
Looks
time
like
now.
most of them are 46,734 seems to be the consensus.
Wonderful. That is absolutely right. And you get that at the end of the trimming report for the reverse read. And so again, the reason it is at the end of the reverse read trimming report is because it had to wait until it did the quality trimming of the forward read and then do the quality trimming of the reverse read. And then it takes a look for each read. It looks at the length of the forward read, the length of the reverse read, and if either one of them are less than 20, it removes both because this is paired end sequencing. So we always need to maintain the same number of forward and reverse reads because all of our downstream tools are going to expect it with paired end data. That brings us to the end of trimming and the end of our first week of the boot camp. I know it's been a lot. I know that we're a little bit behind on the schedule. I do think that we're okay. We're about half a day behind. Remember, we have a full buffer day at the end, so we're still within our spec of time, so no need to worry about that. All of you guys, before you sign off, go ahead and click that floppy disk to save where we are in our Jupyter notebooks. And when we get back on Monday, we're going to start by assessing the quality of our trim data. And if you guys want to export what you've done so far, you can go to File and then Save and Export Notebook As. Save it as an HTML file, and it'll prompt you to pick a place on your local computer to save it, and you can go ahead and save it if you would like. But please make sure you save that floppy disk so that way it'll show up when we log on again next week. And I really, really, really recommend you guys over the weekend to at least review the Unix intro notebook and the R intro notebook if any of that was new to you. It'd be great if you want to go through and do it again. As a reminder, you can just double click and open it up, log onto the system, double click, open it up, and you can go to kernel, restart kernel and clear all outputs, and that'll remove all the outputs so you can go through it again from start to finish. Highly recommend that. I'm going to ask that when you guys are done, and if you guys are logging on over the weekend, which again is great, I encourage you to do so, but just remember when you're done for the day, file and log out. So make sure that you log out or at least go to Hub Control Panel and stop your server when you're done for the day. So with that, we will end this week's session and have a great weekend. Get a lot of rest. Hopefully some of this information will be transferred to your long-term memories. And then any questions you guys have, we will pick up with on Monday. So have a great weekend, and thank you guys for sticking with us for a week. I know that this is a lot.